This is a recording for bioinformatics, BIO5INF, looking at how to use VMD or the visual molecular dynamics. If you go to the bioinformatics LMS page, you will see the worksheet number 5, protein 3D structure VMD. Now, this is a free software. You can download this software on your own personal computers or laptops. It's compatible with both Mac and Windows. So if you go to Google and just Google VMD, just click on the first option, scroll down, download VMD. You can see that there is version 1.9.4, which is the latest alpha. Don't use this one yet. Use 1.9.3. You can see that there is the option for Windows and also Mac OS. Now, I am gonna start the software. So depending if you're using Windows or Mac, I'm using a Mac. However, the instructions are the same. The interface is the same. It does not change. Okay. So I'm gonna keep this display window here and also the VMD main. You don't need this setup command window, so we will just minimize it. All right. Looking at the worksheet, sorry, it's just gonna take a minute to load. Okay. All right. So you will see multiple instructions. It's easy to go through these instructions with a molecule open so that we can manipulate it and everything makes sense. All right. So I'm going to skip the instructions because it's going to be easy when we manipulate the molecule. We'll go straight to the questions. So when it gives you a file code, you need to download this file. And to do so, you just copy the file number or code, go to PDB. You can just search PDB, which stands for Protein Data Bank. We click on the first option. In the search bar, we just paste, search, and we will come up with the molecule. So this is a unique code for each molecule that is entered into the data bank. So we know now that this is hen, chicken, egg white lysozyme molecule. Now, there is some information here. You can see that it comes from the organism, which is the chicken. It is solved through X-ray diffraction to 2 angstrom. And also we have the literature, the publication where this was published. We also have some chains, the sequence length. And also we have some 3D view. It's just a simple way to manipulate the molecule on the website without the use of VMD, but we still want to use VMD. We also have annotations, and this is extra information that will help us render the molecule. So it's chain A, for example, most molecules will have more than one chain. that correspond to a particular complex or sequence. So you can see there's more information in here. We also have the sequence of the protein that was solved that you can get the FASTA sequence of, length, polymer 1, etc. Also have visual representation of what is what, beta strand, alpha helix. Okay, so now we have done this. We'll just go to the main page. Now you need to download the PDB file. This file will contain all the features. Don't put the file number into VMD directly. There is an option to do that. You need to go to PDB and download the file. During an exam or a test, we will not accept an excuse saying that I have entered the number straight into VMD and retrieved the file through VMD. You need to go to the data bank, download the file to your desktop, and open that file. So we'll just go to download, PDB format, save file, and you can see it's a small file and it's very quick. And I'm just gonna put it on my desktop. Okay. Now we need to open the file. I'm just gonna minimize this a little to the side. So we go File, New Molecule, Browse, and I'm gonna go to my desktop and open the file. So once you have located the file, just click on Load, and you will see the file loaded. Okay. All right. Now I don't need this window anymore, so I'm gonna minimize it. However, I need this main menu window, and just to take you through one thing before we start, we can change the background from black to any other color. You just have to go to Graphics and Colors, Display, Background, and you can change to whatever color you want. So usually people change it to white, especially for publication purposes. However, we will stick to black because it gives us a good contrast. Okay. Now to manipulate this molecule, we need to get the graphics and representations. So we need this window. You can see that the molecule has different colors and is in lines. We can manipulate it by changing the coloring method. So we can go to Color ID, and from this drop-down menu, you can change it to whatever color you want. Just leave it as red. We can also change the drawing style or the drawing method from lines to something else. So now if I change the drawing method to new cartoon, you can see the alpha helices and the beta sheets. So I'm rotating the molecule just by left click with my mouse and holding my finger and then just moving the mouse around. Also, if you scroll up, you zoom out, scroll down, you zoom in. Okay, this is just to give you an idea about the orientation, the X, Y, and Z. All right, now let's look at the first question and see what rendering, which means what editing we need to do. So display the molecule in surf rendering without the water molecule. Okay, so now this is in new cartoon and it's only one representation. So I'm gonna create another representation so that I can manipulate the molecule. Now you can have as many representations as you want. It's just that if you want to deactivate one, you double click on it and it will turn red. If I deactivate the other one, you can see it's gone. It's been deactivated. So I'm gonna activate this one and also activate this one. Now the second rendering that I have, I need to only display this molecule as protein without any water. So I'm gonna change the code for this particular representation. So I'm gonna delete all, go to selections. From these single words and keywords, I can choose what codes I want to display. So I want protein and I apply. You can see that now I have changed the selection to only protein. I'm gonna go to the drawing style, change the color to yellow, and change the method, the drawing method, to surf, as the question is asking for surf rendering. I don't need the first representation activated. However, the first representation is of all the molecule. So if I'm gonna change it to surf, see, because this is all molecule, it includes the protein water molecules as well. And that's why you see this overlap between yellow and red. So I'm gonna take this a step further. Deactivate all. You can see that we are missing now some parts in the molecule. So if I activate again, you can see some things appear, deactivate, they disappear. So I'm gonna take this further and create another representation. Go to the color, make it blue, and change now the selection from protein to water and apply. You can see now we know the difference between which part is protein and which part is water, because this representation now has a selection for protein only, and it has a yellow color, and it's in surf. If we look at the other representation, it's only of water, and it's blue, and we can see it's water. So if I deactivate the water, gone. It's only the yellow protein. With water, you can see the water molecules in blue. So the protein is asking for surf rendering without water molecules, which means we only need to have this active. And this is what it looks like. So you can take a screenshot of this and just save it where you want to save it. Okay, let's move on. Question 2. How many disulfide bonds could the protein contain? List them and indicate the residue numbers of the cysteines and that create them. Okay, so in this case, we don't need this representation. I'm going to delete it, and I also don't need this representation. I only need this one. All right, so disulfide bonds form between two cysteine residues, specifically between two sulfur atoms. So each cysteine residue will contain one sulfur atom. So you need two cysteine residues, two sulfur atoms, so that you can have the disulfide bond. So to do so, we're just going to create a new rep. In this new rep, I'm going to leave protein, but I'm going to select element because I am after the sulfur. You need to double click it So you need to see protein element, and then I'm gonna double click on S, and we have S, and I will apply. Now you don't see a difference because the protein representation is active and in surf, so it's masking everything. So if I deactivate protein, you can see these are the sulfur atoms. Now this is not a good representation of the sulfur atoms, so I'm just gonna change the drawing style to bonds, and now we have 4 bonds, which means that there are 8 cysteines. Now to label these cysteines and also to get the length of each of these disulfide bonds, we go to this main window, mouse, label, bonds. Let's do the first one, and I will zoom in. Now I'm gonna, with my cursor, just click once, left click on this end of this tube shape. And I have now cysteine 30. I'm gonna go to the other end and do the same. And now I have cysteine 115. With disulfide bond of 2.42. We do another one. Click on this end and this end, and now we have cysteine 64 with 1680, disulfide bond length of 2.34. We do for this one as well, cysteine 76 with cysteine 94, disulfide bond length 2.22, and we have the last one. Oops. Do the same, and now we have cysteine 6, cysteine 127, disulfide bond of 2.06. So we have all of them now. And also you can tell from the length that the disulfide bond is between 2 to 2.5. So we have answered question 2. We have 4 disulfide bonds. We know between which cysteine. They're all listed. Now, if you're done and you want to delete these labels, you can. All you have to do is go to graphics, labels, and let's start with the atoms, and you can highlight all of them, delete, you can see that they're gone, and then we go to the bonds and we also highlight all 4 and we delete, then we close this window. Okay, so now we don't need these bonds, so I'm going to deactivate this representation and activate the other one, which is the whole protein only that we had deactivated previously. So moving on to question 3, would amino acids 48 to 60 or amino acids 114 to 126 make a better epitope? We need to understand the question before we attempt to answer it. So an epitope is a region of the protein that is recognized by an antibody to bind. Most epitopes are going to be exposed on the surface so that the antibodies can recognize them. In this case, we want to see, is this region 48 to 60 better exposed, or this 114 to 126? To do so, we need to create two more representations. Each will have a different range with a different color. So to do that, we create new representation. We go to the VMD main, extensions, analysis, go all the way to sequence viewer, and then we have the entire sequence of the protein in here. All we have to do is highlight the sequence that we want to manipulate. In this case, we'll start with 48 to 60. I'm just gonna make this a little bigger. 48 to 60. And it created a new representation, which is good. So I don't need this one now, and I'm gonna delete it. Now, here is the selected atoms and the command. We need to change the color and also the drawing method. So let's give this a color red. And because it's in bonds, we can't see it. So we need to change it into the same drawing method as the whole protein, which is surf. And you see now the overlap. Can you see? This is the region now that we have highlighted in the new representation, which is 48 to 60. We need to create a new representation for the other region, which is 114 to 126. So create a new representation. And now it's 114 to 126. So we need to change the color of this one so that we can differentiate. So I'm gonna make this one green. Maybe not the best to make it purple. So you can see now this is the other region 114 to 126, and this is a better epitope because it's more exposed, as you can see, with a larger surface area, considering that this red region 48 to 60 is a small stretch and it's mainly buried. Okay, so now with amino acids this or that, we know that this is a better epitope. Okay, moving on to question 4. Color the 8 hydrophobic amino acids listed here in a different color to the rest of the protein, and then indicate likely to be helices or beta strands. Okay, and we have hints here: new cartoon, exposed or buried. Okay, so now we don't need these representations of the regions. We're done. We can delete them. Delete that one and also delete that one. Now I'm gonna create a new representation of the whole protein, but I'm gonna change the drawing style for the whole protein to new cartoon, and I'll need to deactivate the surf so that I can see the new cartoon. So this is the new cartoon representation of the protein. If I activate the same representation but in a different drawing method in surf, because it's big, it's gonna encompass the whole new cartoon. So I don't need that because I need to look at these residues. Okay, create a new representation. Let's deactivate it for now until we're done manipulating it. I'm gonna change the color of this to blue. Go to selections, and also we now can go to these keywords and find what we need. If you can't find it in these keywords, then you can go to these single words as well. So we just look for a command, hydrophobic or hydrophilic. So we found this hydrophobic, so we can delete this, put hydrophobic, and apply. Now we need to activate, and once we activate, we see all the hydrophobic residues in blue. Now we can create a different representation for each individual amino acid that is hydrophobic. And we will do that in a second. But now to answer this question, we can see that all of these hydrophobic residues are in alpha helices almost exclusively, or in turns. So if we zoom in, you can see alpha helices and also turns here, alpha helices. So we know these amino acids are likely helices. And are they more likely to be exposed or buried? We can check that by changing the drawing style into surf and activating the surf protein representation. So you can see they are barely exposed, so they are hiding inside the protein. So if we deactivate this, you can see that they are inside. And it makes sense because everything that is hydrophobic is hiding away from water. And if you remember, this protein has water molecules in it. Okay, so we have answered the question. However, I just want to show you how we can label each different amino acid a different color. So I'm going to delete this representation, create a new representation, go to selections. Now we want to keep protein. Then we go to keywords here and find res name. Then we have to select them one by one. So if we start with alanine, double click on alanine, apply. Oops. So the reason the command is not executed is because I forgot to double click on res name to get res name. And then I have to double click on alanine, apply. Now we just change the color to blue. You can see alanine now in blue. Okay. We can create a new representation, go to selections, delete alanine. We will find leucine, the second hydrophobic, double click it, apply, but we'll change the color from blue to red, create a new representation, change from leucine to isoleucine, apply, change from red to probably green, create a new representation, again, delete isoleucine, choose methionine, apply, we'll change it to white, etc. So you can do multiple representations, different amino acid has a different color. So it doesn't have to be res name. It can be a particular residue number. So if, for example, I delete this representation, that one, and that one, and you know from the sequence viewer that you want, let's say, amino acid number 50, you can just select amino acid 50 and you change the color of that one to blue. That is your amino acid 50, but it's in a different drawing method. We'll just put it with one that is consistent. And also we don't need this one. Now we see amino acid 50 in blue. Okay, so we have answered question 4. The active site of this protein is found between amino acids 35, 46, 52, and 57. Use whatever rendering and coloring style you wish to clearly show the active site groove. Okay, so in order for us to do that, we need to select for one range between 35 to 46 and also from 52 to 57. So it's similar to how we have selected the epitopes. So I'm just gonna delete this. No, I'm gonna keep them. I will just get my sequence viewer again, extensions and sequence viewer. And now I need 35 to 46. So we have selected it. So this is the first one. And now I'm going to create a new rep and I'm going to change it from that to 52 to 57. Okay, so we have the two representations for both regions in the question. So I don't need the sequence viewer anymore. The best way to look at the groove is probably to use a surf rendering. So if I change the protein to surf and change the others to surf as well, we can see the binding groove in like a V shape. We can make it better by changing this to maybe we'll keep it as surf just so that we can select it unless we change the whole protein into bonds and also we change the other ones into bonds. Okay, maybe see if I can make this new cartoon. So you can play with this as you like, as long as it shows what you need to show. In this case, the V-shaped groove. So we have this V-shaped groove. Okay, so that is for question 5. Moving on to the second part of the worksheet, you can see now that we have a different file code which we need to download. Now, I highly recommend, because VMD is sometimes unstable, to close it if you're going to work with a new molecule and open the software again fresh. So I'm just going to close this and quit. And also make sure that you close this terminal. And then I'm going to open a new VMD. Then I'm going to go and download this file. So again, I'm just going to put it here, go. Oops, need to turn on my Wi-Fi. So I'm going to put the file name, which is that one. Okay, so you can see now this is a different molecule. It's a transcription factor, E2F. And again, it's from human expression system E. coli. We know the literature, so the paper, and also extra information. So for now, I'm just going to download the PDB file, and then we will go through the other information. So if we go to annotations, you can see now we have more chains. Chain A is DNA and RNA binding 3 helical bundle, and also here. So this is the protein. Chain A and B are protein. However, you can see C and D are DNA. See, protein, 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 A and B, C and D are DNA. And also you have the sequence of these from 5' to 3'. So you have A, T, 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 etc. Okay, so because this is a DNA piece, so this is going to be the main strand, 5' to 3', and this is going to be the opposite complementary, even though it's written here as 5' to 3'. Okay, let's see if there's anything else in the sequence tab. Okay, so we can see the sequence of the transcription factor, and also we have the sequence of the DNA chain C and also chain D, and you can get the FASTA file. So if I click on that and I open it, this is the FASTA of that DNA chain. Okay, so it's important to read this information. Okay, so we are done here. We have the file. So we'll just open the file, new molecule again. Remember, you need to download the file, browse, desktop, and now we're working with this one, and we will load. See, it opens in this default lines rendering. Okay, so I don't need this anymore, but I need my graphics representations, which I have here, and I'm gonna change this to color, let's say yellow, and from line into new cartoon. So clearly, once we switch to new cartoon, we can see that there's protein that has these alpha helices, and there's the DNA strands alpha helix. Okay. So when it asks us in question one, how many chains are in the structure? What type of biomolecules are they each? So we know the answer to this question before we even look at the structure, because if we look at this annotations, we know that chain A is a protein, transcription factor E2F4, and also for B, it's a protein, transcription factor DP2, and then we know C and D are DNA. So we answer question 1 from the website. This is to show you that there's a lot of useful information on the website. What are the sequences of chains C and D? Remember, we can actually download the FASTA just by going to this sequence tab, and we scroll down to chain C, open. This is ATTTT, etc. is chain C. That's the FASTA sequence, DNA sequence. And also if we do the same for chain D, we got the complementary. Okay, so we answered this question as well from the website. It's asking you how many tyrosine residues in this structure. 2 tyrosine residues are very close to each other. Which chain are these 2 on? And their numbers and how far apart. Okay, so for this we need to render the actual molecule. So in order to do this, we need to only look at the proteins. So we need to go to chains A and B only that are protein. So create a new rep, and we go to selections, we delete all, and we just click protein and apply. We need to activate the other representation. So now we have protein only. Okay. All right. So we create another representation. This time we leave it protein. We go res name. We double click it. And then we want tyrosine. So we need to find tyrosine. Tyrosine, double click it. Apply. However, because it's still in yellow, we just need to change it to something else like red. So now we have these red tyrosines in the structure. To better visualize these tyrosines, we can deactivate the whole protein and just leave this representation that has the tyrosines. So now we can visualize the tyrosines in there. So you can see there is 4. So we managed to answer the first part. And then the second part, 2 tyrosine residues are very close to each other. Which chains are these 2 on? So these are the 2 that are close to each other. So if we want to label them, we go to mouse, label, atoms, or even bonds. Bonds is better because it will also give us the length between them. So we go tyrosine and we just go close, click on that one somewhere. Maybe we'll change it to label atoms. Nope, okay, yep, this one, tyrosine 124, it's on chain A. So C stands for chain and A stands for which chain. And then I'll try and click on this one. So it might take you a few times until you find the spot. Yeah, I found one. And this one is also tyrosine 90, and it's on chain A as well. So in order for us to get the bond, just need to do the same. And it's not labeling the bond. However, we can go to labels and see we don't have any bonds. I might change the drawing from this into this. Yep. So we change the drawing to bonds. And I'm just going to click on this end here and also here. So they are 3.53 apart. So we answered the chains, what residue numbers, 124 and also 90. How far apart? 3.53. Okay, so you can see that you can manipulate this. So we used to have the new cartoon and then we change this into bonds. It's just trial and error. Okay, question 4. Could a disulfide bond form between the two protein subunits as shown in this structure? So in order for us to have a disulfide bond, we actually need at least 2 cysteine residues, 2 sulfur atoms. So I'm gonna change this representation. Actually, I'll just delete the labels. So delete the bonds and also delete these. Then I will delete tyrosine and put cysteine. There is only 1 cysteine, which means that we can't have a disulfide bond. If you're unsure, you can always change to surf to see we only have residue 1. New cartoon, there's only 1 residue, which means that we cannot have a disulfide bond. Okay, question 5. Which 5 arginine residues are most likely to contribute to the DNA binding activity of this complex? Okay. So in order to do this, we will have the protein and we will delete this representation. Actually, for this question, maybe we'll answer question 6 and then go back to 5. It just makes it easier. So we need for question 6 to show the DNA and also the protein in the appropriate rendering. So I'm gonna create another rep, go to selection, delete protein. There should be nucleic acid somewhere. Nucleic, and we have nucleic, which is nucleic acid DNA. We can also select as a chain. So if I deactivate this, create a new, we can go all and find chain, and we want chain C and chain D. You can do them together or one just to show you that it can be individually. We'll just do C. You can see only half of the DNA alpha helix is showing. We can create another rep and select chain D, apply, and then we have both. However, it's easy because we know that it's DNA to just click on nucleic. So I'm going to delete this, I'm going to delete that, I'm going to activate this, and I'm going to make it red. So you can see it's in the new cartoon. This is the best rendering of DNA in new cartoon. If you do it in surf, it does not make sense. It's easier to have it. Even bonds is okay. It's not the best. Let's say ribbon. Ribbon is also okay. New ribbons is also okay. However, new cartoon is the best. So I'm gonna make it maybe blue. So DNA is blue. And we have protein in yellow. If we make this like a real protein in surf, this is what it looks like. It looks nice. So you can see it looks like a protein transcription factor bound to a DNA molecule. Okay, so we stop it, and you can capture this image and put it in your worksheet somewhere. Now we'll go back to question 5. Which 5 arginine residues? So to start, we need to create a new rep, and in that new rep, identify the residues, the arginine residues. So we go protein, res name, and arginine, ARG, apply, change the color to red, and there you have it. So we have these arginines here, but it's really hard to select them and know what they are. So the best way to do it is to change from surf back into new cartoon, and also the protein back to new cartoon, and we have them. So we select those that are closest to the DNA binding activity. So the closest are here. So if I deactivate the protein and I also label atoms, so this one is not arginine. I'm gonna have to delete it. Delete, delete, delete. Probably the best way, since now we know that these are the arginines, the 5, so there's 2 here, 1 there, and then there's 2 here, is to probably deactivate the protein and change this into maybe tube or beads, too big. So you can see there's 5, these are the 5, arginine 72, and the only reason we know is because we just deactivated the protein, and we know that these are the ones that contribute. Can you see? We have highlighted them, 1, 2, 3, 4, 5. So in order for you to see them, I'll just deactivate all of these. So it's arginine 72, arginine 122, and arginine 121, arginine 57, and arginine 56. So there you have it. We have answered all these questions using VMD. I highly recommend that you attempt these questions. Again, listen to this recording as many times as you need to, because you need to be comfortable with this program for the exam and also for your case studies. Again, VMD is not perfect. If it acts up, close the program, load again, open it again, and also always download the PDB file onto your computer and open that file. And don't forget that